Well, thank you very much for the organizers. It's a great meeting. Thank you for Ben and Shirley for great communication with me. Everything was super clear. The instructions were great. So my lab, as was mentioned, works with nucleic acid nanoparticles, NAMPs. So I'm going to use this acronym a lot. So whenever I say NAMPs, I mean nucleic acid nanoparticles. So today I'll share just some of our most recent advantages, most recent developments within the field. So in traditional small molecule drug, we have two primary components, the dynaphore, which governs the distribution and delivery of the drug, and the pharmacore, which dictates targeted function. Therefore, any changes in chemical composition of the drug affects both dynaphore and pharmacore component, and consequently, these drugs, these new molecules will have to be clinically re-evaluated at every stage of their development. Informational drugs, such as therapeutic nucleic acids, however, have an independent dynaphore and pharmacore components, which are determined by the backbone chemistry of the nucleic acids and the sequences, respectively. So this improved modularity provides much better predictions for the clinical pipeline of the therapeutics. So here on the right side, I'm showing some examples of mechanisms of nucleic acid therapies. And again, due to the independence of dynaphore and pharmacore components, the backbone chemistry and the targeting with the sequence can be changed, and drugs can be easily adapted to new targets, therapeutic needs. And even more exciting is that with our current advances in understanding of RNA folding and structure-function relationships, we can take one step further in this concept and introduce next generation of therapeutic nucleic acids that are based on nucleic acid nanoparticles, on NAMPs, as I mentioned at the beginning. In this approach, NAMP architecture would be defined as the dynaphore, and therapeutic nucleic acids, or TNA, added to the NAMP would be responsible for the pharmacore function. So this innovative concept offers some additional advantages in precisely regulating, for example, biodistribution of those molecules, their cell compartmentalization and immune recognition, as I will show later on today. How do we build NAMPs? So we learn from nature and plethora of available RNA structures to identify a set of structural and long-range interacting RNA motifs. And we use them as building blocks, building units to assemble different NAMPs. You can think about it similarly to the Lego blocks. And this process can be carried out manually or aided by computer-assisted tools. For the computer-assisted tools, we need to choose from a set of motifs. For example, in this case, we have two-way junctions that have 120 degrees angle between the adjacent helices. And if we place 6 of them together and then identify an appropriate spacing between them, we can form a closed three-dimensional structures. So once we define geometrical parameters of those structures, we can optimize the sequences and define the connectivities, and we'll have ready-to-go NAMP. Another approach would avoid the use of any structural or long-range interaction motifs and rely solely on intermolecular interactions without any intramolecular bonds. So there are no secondary structures within each bond. And this strategy includes computational sequence optimization, which involves normally sequence randomization and then some sort of Monte Carlo optimization algorithms of that. So by using those two approaches, we can create a plethora of different nucleic acid nanoparticles, or NAMPs. And once appropriate dynaphore NAMP is selected, we can add different therapeutic nucleic acids to define the pharmacore function of the formulation. And again, we can tap into the diverse functional diversity of nucleic acids to modify NAMPs, and then produce nanoparticles will have some therapeutic meaning in them, and would be able to deliver a cocktail of different therapies into the same disease cells in a very synchronized and precise nature. The application of these technologies have been successfully demonstrated in various animal models and have been also licensed by some companies. NAMP technologies offer several advantages. Some of them are listed here on the right. And when delivering a cocktail of therapeutic nucleic acids, different NAMPs architectures can be utilized. For example, we can use cube-like nucleic acid nanoparticles to deliver 6 different siRNAs into the same cell. And then they are released via the dicer-assisted release will provide the targets and simultaneous targeting of 6 different genes within the same disease cells. But the same therapeutic cocktail can be delivered by another NAMP, such as, for example, nanoring here. So this versatility brings a question: what do cells see when different nucleic acid nanoparticles are introduced? And that's a very good question, because the immune cells are equipped with the extensive portfolio of so-called pattern recognition receptors that detect foreign nucleic acids. And those PRRs, or pattern recognition receptors, can be present both in cytoplasm and also endosomes as different lines of defense. And this detection of foreign nucleic acid materials will activate a complex network of signaling cascades that usually culminates in the production of interferons or different types of cytokines or chemokines. And while controlled immunostimulation may be beneficial and have some beneficial therapeutic effects, the unregulated induction of cytokines may lead to some undesirable inflammation and cytokine storms. Therefore, it becomes crucial to understand the immunorecognition of nucleic acid nanoparticles, NAMPs. So in close collaboration with Dr. Marina Dobrovolska from Nanotechnology Characterization Laboratory, we initiated the very first systematic studies of NAMP immunorecognition. We carefully selected a library of NAMPs and analyzed them all to address the influence of NAMPs dimensionality, composition, sequence, connectivity, and size on their immunorecognition in human peripheral blood mononuclear cells, PBMCs. We chose PBMCs because it was shown to be more reliable as the model in prediction of cytokine storms and various toxicities, and this model is more accurate than common preclinical animal models, which include, for example, rodents and non-human primates. Our work was well received and recognized by various news outlets, and was also featured on the cover of the NCI Congressional Report of last year. You can see on this cover the PBMCs with the green dots inside. So these green dots are fluorescently labeled NAMPs uptaken by the PBMCs. So over the course of several years, we developed a very robust experimental procedure that details the preparation of endotoxin-free NAMPs, isolation of PBMCs from freshly collected human blood, and analysis of characteristic biomarkers produced by PBMC in response of nucleic acid nanoparticles. The continuous execution of this protocol is about 48 hours, and when the immediate analysis is not feasible, single-use aliquots of supernatants can be frozen and stored. This protocol is now publicly available and can be used by anyone in the world. By following this protocol, several important discoveries were made. We assessed the contributions of NAMPs architectures, compositions, and their functionalizations on the primary contribution to the immune recognition. For example, globular structures on average were shown to be way more immunostimulatory than fiber-like structures, and nanoparticles made solely of RNA are more immunostimulatory than the same nanoparticles made of DNA, and so forth. We also identified that without carrier, nucleic acid nanoparticles are completely immunoquiescent. Because of their size, they cannot enter the cells. And this makes them perfect candidates for various biomedical applications that require extracellular use of the material. For example, we can build some monoclonal antibody mimics using nucleic acid nanoparticles. We also identified that plasmacytoid dendritic cells are the main responders to the nucleic acid nanoparticles, and we introduced some quantitative structure-activity relationship models that allowed us to determine the main contributing factors in physicochemical characteristics of the nucleic acid nanoparticles in relation to the immunostimulation. I will talk a little bit more about this later. This work was done in collaboration with Alexey Zakharov from NCATS. We further studied the effect of altering the chemical composition of NAMPs that have the same connectivity and shape and size. So, for example, if you look at those nanoparticles, they all look like triangles and they're all made of 4 strands, but those strands can be either RNA or DNA or 2' fluorinated RNAs. And we can assess the homogeneity and morphology using atomic force microscopy, confirm the assembly via different gel electrophoresis techniques. What we discovered is that replacing RNA strands with either DNA or chemical analogs strands increases the enzymatic and thermodynamic stability of nanoparticles, so we can precisely fine-tune the physicochemical characterizations. And we also see no effect on integrity upon release of those nanoparticles from lipid-based carriers, as shown in the gel in panel D. This work was done in collaboration with Emil Khizmudinov from Ball State University. We discovered that the same architectures, like as I mentioned, 4-stranded triangles, but different compositions are recognized differently by the immune cells. So here are the responses to those nanoparticles introduced to human microglia cells, for example. And you can see that depending on the nanoparticles composition, the responses are different. Also, the compartmentalization of those nanoparticles within the cells differ because based on their composition. We can either assess it using the microscopy tools, or we can do fractionation experiments where we basically disassemble, break cells apart, and then see the presence of fluorescently labeled nanoparticles in different fractions of the cells. So to summarize, we discovered that depending on composition, our nanoparticles can be either immunostimulatory or immunoquiescent, that they can either go into the nucleus or stay in the cytoplasm. Or if they stay in cytoplasm, they can be recognized by different pattern recognition receptors, for example, by RIG-I, or they can be recognized by RNA pol-3, polymerase 3, to produce RNA that is then recognized by RIG-I. So that's a very highly versatile and tunable platform that we have. We also assessed the influence of the structure of the nanoparticles due to the modular nature of NAMs by assembling different combinations of functionalized and non-functionalized monomers. We can create a library of functional nucleic acid nanoparticles with regulated spatial orientations of therapeutic nucleic acids that we then can confirm by using different techniques and analyze in cell cultures or in animals or in the blood of the healthy human donors. What we discovered that, yes, all the nanoparticles that we predicted to form are being formed. You can see it from the atomic force microscopy images or from SAX data, small angle X-ray scattering. We discovered that depending on the orientation and number of functional moieties attached to the NAM, the immune recognition changes and different pattern recognition receptors participate in their recognition. And this is not only true in cell culture, but also in animal studies. We showed that orientation of the therapeutic nucleic acids affects the in vivo response of the NAMs. We did some in vivo imaging, ex vivo imaging. We identified that depending on the composition, having the same carrier, the delivery of our NAMs to different organs changes. And so does the immune recognition of these particles in vivo. So when we assess the immune recognition and PBMCs collected from different donors, we saw very nice panels which allow us to identify a suitable drug delivery platform for a particular patient based on the composition of this platform. For example, for donor 3 or for the patient 3, formulation 4B would be optimal because it doesn't elicit any immune response at all. So it's a very quick experiment which can be done at the bedside, and then based on the response, we can define which composition of the therapeutic formulation we should move on with. So for a quick summary from what I just told you, we have identified several endosomal and cytosolic signaling pathways essential for immune recognition of our nucleic acid nanoparticles. They can be, and those pathways can be precisely targeted via tuning nucleic acid nanoparticle structure and composition. We also recently introduced a new approach that allows you to go from immunostimulatory to non-immunostimulatory nanoparticles through their passivating with the therapeutic moieties. For example, if we take fiber-like structures and introduce them into the human cells, they can be activated with, recognized via the SIGA STING pathway because SIGAs can easily recognize those fibers and bind to them, as can be seen here on the predicted models. However, if we put some therapeutic moieties on those particles, depending on the density and orientation of those therapeutic moieties, they can shield the NAMs from being recognized by CGAS, and therefore those NAMs becomes undetected. So, as I just demonstrated you, the incorporation of therapeutic modalities while maintaining a particular level of immune stimulation of NAMs requires a feedback from the experimental analysis to the rational design of nucleic acid nanoparticles. In other words, we have to come up with the design of nanoparticle, then synthesize it, which may take several weeks, characterize it, which can take another week or so, and then do extensive characterization on PBMC, which may take several days if we have access to all the materials. So the question is, can we somehow speed up this process? So we started this collaboration with Alex Zakharov and Marina Dobrovolskaya several years ago. And in the course of the past 5 years, we developed an unprecedented computational tool so we developed this computational tool that we call AI cell, like artificial intelligence cells, but it also stands for artificial immune cell. That allows us to accurately predict the immunological properties of any nucleic acid nanoparticles designed just based on the sequence input. So this work combines extensive experimental work, which was then fed on the different computational algorithms to come up with the best approach to predict the structure, best predictive model to assess the structure of nucleic acid nanoparticles. This platform right now is publicly available. Anybody can access it on the website. When you access this AI cell, you just need to click the predict button, and it will compare the new nanoparticle that you are trying to assess against all the nanoparticles that we currently have in the database. Right now, it's about 70 different nanoparticles, and currently we are putting more, about 200 nanoparticles, to expand the possibility of the program. And then it will allow you to input the number of strands which enter the composition of your nanoparticles, and you can specify it. Here, for example, it's set for 4 strands, and once you put them in, you click predict button, and only after 2 to 4 seconds later, you get the estimates for the production of type 1 and type 3 interferons, which are the most reliable cytokines for prediction of immune stimulation of nucleic acid nanoparticles. So this approach is much faster, of course, than doing it experimentally, and at least it can give you some hint on whether or not you should proceed with a certain formulation. So this tool can be found at this website, and this work was published also in Small a year ago or so. Please use it, give us your feedback, and based on your feedback, we'll continue it even further. So right now we're working on addressing the CNS diseases, and we use the appropriate cell models to do this. With this, I would like to thank all my collaborators who contributed to this massive effort, as well as my lab members and alumni from the lab. I would also like to thank the sponsors, NIH and NSF, for the generous support of this project. Thank you very much.